Colloquium. Today our speaker is Jordi Soriano Fradera from University of Barcelona. As usual, I'm going to introduce a little bit our speaker before starting. So, Jordi got his PhD in 2003 from University of Barcelona, and then he had a few postdoctoral appointments, one in Germany, one in Israel, and then he moved back to Barcelona, first with a prestigious fellowship, Spanish fellowship, the Ramon y Cajal Fellowship, and then he became a faculty in University of Barcelona. He's an experimental, and his research uses neuronal cultures as model systems for both fundamental neuroscience and medical applications. He has a laboratory that develops neuroengineering methods, imaging technologies, and data analysis tools that place brain-on-chip approaches at the forefront of research in Catalonia. And over the past years, this team has advanced our understanding of neural network dynamics and contributed to new therapeutic strategies for the Parkinson's disease, for example. A key achievement include that they've been able to demonstrate the role of modular organization in neural circuit function and identifying astrocyte dysfunction as a driver of genetic Parkinson's disease. This has also shown that genetic therapies can reverse neuronal damage, and this opened the window to new possible therapies for this disease. So with this, I shut up, and I leave the floor to our speaker. Thanks.
Thank you. Thank you very much. Thank you very much, everyone. Thank you very much, Silvio, for organizing this event and inviting me here. It's a pleasure to be back to Sao Paulo and meet all the people and discuss the different ideas. So today I will provide an overview of the neural cultures. And I would try to convince you that they are a very nice model system for physicists, for medical people. They provide complementary approaches to what we have seen, for instance, with Mario at the level of the brain. They are very useful toys. You can play a lot with them. And I just wanted to show you how nice they are. Hopefully you like them. Okay. So I'm actually experimental physicist and I'm physics. I studied physics, but I have a lab in neuroscience in Barcelona. So most of my efforts and sometimes my pain, my nightmares is to try that to make my neural cultures to work, okay? Good, so before going into the definition of what is a neural culture, let me just frame my research a little bit. And it's because now there is a lot of interest around the world to do something at the level of in vitro systems like neural cultures and try to see if they are intelligent or not, okay? This is what people call in vitro intelligence. And the idea is that of this, and actually I put part one, because I will finish my talk also going back to this problem, okay? But you need a little bit of, let's say, overview to understand what can one do with this in vitro intelligence. So the idea is that for about 4 years, I worked in this European project in which we wanted to basically emulate artificial intelligence in biological circuits in vitro. The idea was to take neurons normally from cell lines or from rodents, and then put them in like chambers like this, okay? And the idea is to emulate key principles of the organization of the brain, like modularity, and in principle, we wanted to emulate modularity and other aspects, or try to have something that is called brain-on-a-chip system, then apply stimulations to somehow dictate the connectivity of the system. And in principle, if everything works and you're going to use plasticity and tune your network, when you apply an input, the system is able to recognize that input as something, in this case, like a number, okay? Which would be the super basic idea for classification of digits, these kind of things. And people consider that if you are able to really teach a neuronal network in vitro, okay, for recognition, for pattern recognitions, maybe also to move robots, all these things, then you are advancing in the understanding of how basic intelligence works in vitro, okay? This is a challenge that people like very much, for instance, for the development of implants. That is very, very important. And the problem is that this was a beautiful problem, but it was very, very difficult. So the idea was to design and train neurons grown in the lab in this kind of system, and then blend electronic and biological systems so they can talk to one another. So in principle, if something is wrong with my biological network, the electronic one can take care of that and help the biological one, and in principle, the opposite. If the electronic system is a robot or an implant, then the biological network can help the robot to operate correctly. It's a beautiful problem. It's a very nice idea, but as I said, it was very, very difficult. But the fascinating thing is that one of the aspects was modularity. People is really obsessed in trying to imprint modularity aspects in vitro systems. And the reason is that the brain is modular. So basically, different functions are basically dictated or governed by different regions in the brain. For instance, this is the motor cortex. So each different parts of this motor cortex govern the movement of a particular muscle. And so the brain operates in this way. We have different cortical areas, for instance. These are the high thinking areas. This is the visual cortex, and so on. So these different areas process information separately, and then information is put together to understand the environment and take decisions. So modularity is so important, so much present in the brain that people that wants to do something in vitro that resembles the brain is always thinking about modularity. And to make a little bit of joke about myself, actually, I just wanted to show this video because someone of the audience asked me to put this video. And this is me 25 years ago, okay? It's the same Jordi. But this is an example of how the motor cortex works, because the idea is that I'm there, and this guy in the back, this colleague, will use a magnetic field to stimulate exactly this finger, okay? So we'll be moving this finger, and my colleague will stimulate the finger and create a perturbation. So it's just to show you how precisely you can target a specific cortical circuits. Yeah, I'm here, I'm alive, so don't get scared. So this is the coil for the magnetic field, and my colleague will target exactly the cortical area that controls my finger. So my finger is doing what is called finger tapping, and now a perturbation will be applied. My finger will get completely crazy, okay? And will follow the rhythm of the magnetic field imposed by the coil, okay? So this is just an example, a funny... Correct, yes. You have to use a very strong magnetic field because you have to pass 1 centimeter of a skull. So basically, you apply a few Teslas of magnetic field to get few Gauss in some neurons that are those that activate, okay? Sorry?
Unless you decided to continue with this job.
Yes.
You were not scared, let's say. No, okay.
It's fun. It's really interesting. No, but the reason of doing this is that this was the beginning 25 years ago of a therapy that now is transcranial magnetic stimulation that is used actually to help people in different diseases, okay? We can go into details later if you want during the coffee break. But anyway, the point is that modularity is important, it's there in the brain, and that's why people want to imprint it as best as possible in systems in vitro, okay? Yeah, so then we were here, and then what we learned after 4 or 5 years of work in this problem is that it's extremely difficult that I put neurons in vitro and I wire them as I wish. It's extremely complicated, okay? So one cannot Lego at will, let's say, the connectivity of a neural network. However, I learned... No. This would be awesome. This would be ideal. But it's very difficult. People is doing that. There are printers for neurons. And people is trying to really Lego to make circuits precisely wired, but it's extremely difficult. But there is a lot of money on that. And the reason is that, yeah, there is a lot of interest in understanding how the system works, let's say, bottom up. Yeah, this is the basic idea. And if the system is able to show intelligence, even better, okay? So the point is that it didn't work very well, but I learned a lot of things. I exchanged a lot of ideas with colleagues around the world, and now these kind of initiatives are present in the United States, also in Europe and in Japan with this multicellular neurobio-computing, which sounds very spectacular, okay? And actually, there is a company in Australia called Cortical Labs, where they actually teach neurons to play video games. And this is how I will end my talk today. But you have to wait, okay? Anyway, but the message is that all these ideas of building systems in vitro and trying to show them or play with them to show intelligence is something that is a very hot topic today. Okay, good. So now I can introduce the neuronal cultures. Neuronal cultures are normally prepared from cell lines, human cell lines, or rat mouse neurons. Basically, you buy a rat. The rat is about €25 or dollars, something like that. They're cheap. Then you dissociate the neurons. You put the neurons on a glass cover slip together with nutrients and resources that the neurons need. And typically, you have to imagine that neurons grow in this kind of wells that you have here. Okay, it's just for you to get an impression of the scale. Okay, and you can put there about 50,000 neurons in this well, 50,000 dissociated neurons. And I'm playing this video here for you to observe that they very quickly they extend connections, they wire to one another, and basically in few days you get a network that is extremely wired. Okay, and so typically after 4 or 5 days, the system is able to exhibit what is called a spontaneous activity. I mean, without doing anything, the system is able to activate itself, which is what I'm illustrating here. This is a spontaneous activity taken in my lab with a technology called fluorescence calcium imaging, in which basically I use a fluorescence probe that binds calcium, and calcium is one of the ions that is exchanged during neuronal firing. So in principle, it's relatively easy to detect the activations of neurons. So you prepare the cultures, you keep them healthy in this kind of wells with nutrients and so on. They self-organize again. Okay, not only they wire, but they self-organize to be able to exhibit a spontaneous activity. And no matter what you do, you have to basically kill the culture to stop that spontaneous activity. And this is very important. And it's very important because in the brain, during the formation of the retina, for instance, or the spinal cord, those systems start to wire themselves and show a spontaneous activity, and they need that to function properly when we are born. So spontaneous activity is not a joke or something to play around for physicists. It's something extremely important to understand. Yeah, but remember, the system is able to activate by itself. Okay? And just to make things clear, I want to just emphasize how neurons work. So neurons receive inputs via these dendrites that can be excitatory, that increase the memory potential, or inhibitory, that reduce the membrane potential. Okay? And conceptually, you need the quorum, meaning that you need a sufficient amount of inputs in total in a short-term window for the sum to pass a threshold, and then the neuron will fire and will go back to the resting state. Okay? This is conceptually the idea, and this is the intrinsic nonlinear behavior of integrate, get inputs, and fire, pass an input to other neurons. Okay? This is the conceptual way for the neurons to work. This signal here reaches the end of the synaptic cleft. The electrical signal is converted into a chemical one. The information passes via neurotransmitters. Then this chemical signal is translated again into electrical signal that corresponds to the events that are arriving into the dendrites. Okay? How many neurons do you need for this self-activation? Oh, that's a good question. Actually, you can do it with very few neurons, with 10, 100. It's a good question because if neurons will enforce the connections, okay? So with few neurons, they will be able to show enough drive to show a spontaneous activity. Meaning that if I have maybe 2 neurons, the connection will be extremely strong. If I have 100, they will be weaker connections, but altogether, the total balance is that there is enough drive for the system to spontaneously activate, okay? Yeah, and it's also a good question because not only the electrical signal is converted into a chemical one, okay, to pass the information to another neuron, okay, but also these neurotransmitters here are released spontaneously in very tiny amounts, okay? And this is very important because for us as physicists, these tiny amounts of neurotransmitters go into this postsynaptic area and they cause a spike, cause an activation of the neuron. This means that you have a natural source for noise, okay, which is something that we like very much in physics. Yeah, so we have intrinsic dynamics of the system of the neurons, and also we have information propagation together with noise. Okay, and this is very important, and this noise is actually what facilitates this spontaneous activity. And the third thing that is important is the network itself. Neurons are not isolated, of course; they are wired. And then you have to take into account that we have excitatory neurons that are marked in blue, inhibitory ones that are ones in red, and also other cells that are called glia that support the system and they ensure that neurons are healthy all the time. Okay? Something very important is that excitation and inhibition have to be balanced, and in our real brains, they are all the time changing, okay, to control the traffic of information, okay, and process meaningful information. But so you have to imagine that the balance between excitation and inhibition is important, like a traffic light in a city. Okay, you need a reasonable flow of information, yeah, for the concepts to be transmitted across the network. So the important thing for me is that I can play. In the laboratory, I can put drugs that alter the behavior of the neurons. I can put drugs that alter the communication and the noise of the system. And of course, I can do different strategies for me to alter the connectivity of the network. And that's why neuronal cultures are very beautiful, because I can play with all these three systems with relatively, let's say, minor ethical constraints, okay? I can play a lot, and the police will not come to say, okay, what the hell are you doing, guy, okay? Yeah, I cannot do these things with humans, as Mario knows very well, okay? Good, so the problem is that when I play with neuronal cultures, they're very beautiful, they work very well, you can keep them healthy for, let's say, 2-3 months easily, okay? It's very nice. They are relatively cheap, and the problem is that if I look at the spontaneous activity that I was mentioning before, the spontaneous activity is like this, okay? So all neurons activate together or die silent, activate together or die silent. It's a very boring behavior. It's not synchronization. This is a very fast propagating wave, very fast, about 50 millimeters per second. It's a fast wave, but here, in this movie, it looks like synchronization. The point is that the system is locked in a supercritical scenario in which everything or nothing, okay? Very, very boring. But if I look at the spontaneous activity in the actual mouse brain, you see I have a very rich repertoire of activity patterns, okay? This is the same thing, spontaneous activity, no inputs from the outside, the system is just activating itself. And the point is that you can see here that there is a huge exploration of possible activity patterns. This means that this system is very rich in behavior, and this one is very poor in behavior. So the first thing that caught the attention of my lab is what do I have to do in the laboratory so these neurons in culture behave somehow like these ones. And if I manage to do that, then I have a system that is much more interesting, let's say for the medical community or for the physics community, because I have something that at least from a dynamical point of view is closer to the brain. It's not a brain. Don't say that I'm making brains in the lab, okay? It's not a brain, but from a statistical point of view, it is like a brain, okay? And this is what I want to achieve. A system like this, which will help me not only in the, let's say, in vitro intelligence part, but also in having systems that for the medical community are very, very interesting. And to play with that, here I put examples of what we can do. One is to take, well, this is our reference, a homogeneous network. Then I can put, for instance, then the neurons making this kind of aggregates. So they are like modular structures by themselves, but this system self-organizes. No, there is no engineering there. So just put neurons in small spheres, and it self-organizes. But then if I want to be more refined, I can put neurons in particular traps. Okay, so it's an engineered modular system, but I control precisely where neurons are located and how they connect, okay? So modular, but this is self-organized and this is engineered. And then I can do more crazy things like putting traps or topographical patterns, so neurons have to follow intricate paths to connect to one another, and this makes the system much more richer. Actually, to show you these ideas, I will now pass. You can now touch the toys I brought from the lab, okay? So you can get an impression of these topographical patterns, how they look like, okay? So you can pass it. If anyone, after my talk, if anyone falls in love with these kind of patterns, you can take it. But please tell me because I have more, okay? So the point is that with these topographical patterns, you can really enrich the connectivity of your system. This would be the idea, yeah? Okay, good. So here I put a repertoire of the different videos. This is my reference system, okay, the homogeneous isotropic growth of neurons in my network. The next one is what I said is the self-organized modular system. And as you can see, it doesn't activate anymore as an on-off system, but it's more actually divided, more modular actually in dynamics. This group here activates quite often together. And this is nice because it's already something that goes beyond my very simple scenario. And then here is the engineered modular network. Okay, this is also cute that I have activity sometimes in neurons, sometimes in specific modules, and sometimes all together. So it's also very rich. And this actually, on the fourth one, I'll keep it as a secret for later. Okay, because it's the super beautiful one. Okay, good. So just for clarity, I can do in the lab. I can apply two different techniques to record activity. One is what I was mentioning just now, the fluorescence calcium imaging, in which basically I use indicators for calcium that are fluorescent. So with a simple microscope, I can record the activity of my neurons, as one can see. Oops, as one can see here. If the video wants to play, yeah, as one can see here. Okay. Then I identify my neurons as regions of interest, and I can extract the behavior of my neurons. There is an option, and another option is to use multi-electrode arrays, in which basically I have these things. Okay. These things are electrodes. So here there are 4,000 electrodes. So I can record precisely the electrical signal of my neurons. Okay, they are completely different systems. Yeah, and they are completely different systems. One gives me a relatively poor time resolution. I can take another one. Okay, one gives me relatively poor resolution because the dynamics of the neuron is coupled with the kinetics of the indicator. Okay, so I have about millisecond resolution. But I see exactly where neurons are. Okay, this is very important for me. I know exactly the location of the neurons. The other gives me very nice microsecond resolution. The problem is that I don't know where are the neurons because the system is completely opaque. Okay, so depending on the problem that you have at hand, you go into one technology or the other. Yeah, and in my lab we combine both. The advantage of this system is that I can read electrical activity, but I can also stimulate my system. And this is very nice because I can apply stimulation, try to induce some training, see the response, and so on. Yeah. Okay, good. And then, of course, you have to get the data. For the calcium imaging data, you have these jumps in fluorescence, okay, that indicates that neuron is activating, and then you convert these jumps into what is called the raster plot. So you have to imagine that for each jump is converted into one of these black dots over there. Look that the scale is different, okay? This I wanted to illustrate here the fluorescence signal and here the entire behavior of the network when I converted the fluorescence into spike trains. Yeah? So remember, each of these dots is the activation of a neuron. And this is my data set that I later can analyze to get information about the behavior of my network. So for instance, I can do a lot of things. One thing I can do that is relatively simple is just to look at the dynamics of my system. Yeah? For instance, I can put those spiking events into the layout, the physical layout of my system, and I can observe, for instance, how the propagation of activity fronts take place. And actually, this is very nice because then I can use this information to model the initiation of the spontaneous activity that I was referring before. And of course, I can use this data and then apply pairwise correlations or transfer entropy or many other possible strategies to infer the communication between the neurons. And I can go from this raster plot to this connectivity, to this network and associated connectivity matrix. And if I'm able to compare healthy and disease systems, ideally, I will see something different in the communication of the neurons by comparing the networks. Okay, that is basically a super quick overview of the data flow in a typical laboratory. Okay. Okay, with this at hand, in case that you like all these ideas, I recommend you to take a look to this review of myself that published about two years ago, in which here I show all the different things that we can do within our cultures. Okay, so we can do, for instance, injury. We can apply damage into the culture and see if the system is able to recover or not. You will see an example soon. We can do numerical simulations inspired on the experimental data. We can do complex networks analysis and play with them. Okay, we can do neuroengineering, playing neurons in particular locations, and we can do a lot of medicine in which we help medical people to develop therapies, starting first in vitro. Yeah, imagine that I have neurons that have a genetic disease. So in principle, if the genetic diseases cause damage in the connectivity of the network or in the intrinsic dynamics of my neurons, I should see that in my raster plot and therefore in my network. And in principle, I could talk to medical people to see whether I can use drugs or genetic treatments to treat my diseased culture. If that works, then I can provide ideas for the medical people to later on go into real patients and help them. Okay, that is the idea. Of course, the time frame is decades, yeah, but we are trying to help in this direction. And the numbers that you see here are actually papers from my lab, okay, related to these different problems. Yeah. Okay, good. So this is what we do in my laboratory. And now, very quickly, I want to discuss a little bit the concept of initiation and propagation of activity. So remember that this system is able to spontaneously activate, okay, like this, Yeah, and one of the big questions that many people had is why the system is able to do that. Okay, and we explored this idea both experimentally and with numerical simulations. And numerical simulations are a lot of fun because you can really play with the architecture of your neuronal circuit. And one of the first things that you have to take into account is that for a biological system, for a biological neural network, to build axons, to build connections is something extremely expensive. Okay, it's very, very expensive. You have to keep that axon healthy. So normally, neurons tend to connect in the vicinity, okay, with the minimum wiring. And normally, you make long-range connections. You may have them, but are with very low probability. And normally in the brain, they appear when they are necessary. Okay? Ideally, one would expect the brain to be fully connected, and then you switch on and off some connections depending on the need, right? But that is unaffordable. Okay? So in the brain, we have long-range connections only when they are important. So with these ideas in mind, in vitro, the same thing happens. Basically, neurons connect in the vicinity, and you have, with some probability, some long-range connections. And let me show you an example of how you build these networks in vitro. So you put first the neurons on a surface, then you simulate the growth of the axons, okay, using biologically realistic ideas. And then around each of the neurons that are these bodies, then you will see now that I put what is called the dendrite arbor. So the concept is that when an axon crosses the dendritic tree, a connection is made, okay, from the neuron of the axon to this one, okay? And basically, this is the place where inputs arrive, okay? So with all these ideas, I can finally get a network, okay? And the beautiful thing of this is that when I build the networks like this, because I have normally preferential connectivity in neighborhoods, it's very easy for me to generate triangles. Triangles means motifs in my network, okay, that are special. That, for instance, in this case, for three neurons, this is called feed-forward because this neuron, when activates this one and indirectly activates also this one, okay? So propagation of information is amplified with this feed-forward loop. But this one in feed-backward, you see you enter in a loop that basically closes itself, so information there is somehow retained in my network. And conceptually, the point is that from a mathematical point of view, I can classify how many triangles, how many motifs do I have in my network, and try to understand their importance in the dynamics of my system. Because these feed-forward loops help me to amplify. So the noise I was talking about before, it is the one that helps me to amplify information across the network. And if I reach some kind of critical, let's say, level of excitation in my network, the system may be able to activate. Okay? That is conceptually the idea. And before going forward, let me just show you that one can do a lot of numerical simulations, okay? And this is another example, okay? One can really simulate everything. If you have enough patience and the skill of students, you can really go into the details and try to simulate everything. Yeah, it's a very nice problem. But to understand the initiation of activity, what we observed is that there are two mechanisms that are very important. One is called the dynamic noise amplification, in which if I don't have anything to amplify my activity, I need in principle about 15 inputs. This is the quorum I was mentioning before. This is the amount of inputs that I need for my neurons to arrive in a particular short time window for my neuron to get activated. Okay, so this is called quorum. So all these neurons have to project to this one, and this one needs about 15 inputs to fire. But if I have noise, which is what I wanted to represent here, if I have noise, which are these red bars, then this noise naturally is helping me to activate without the need of so many friends, okay, send me inputs all the time. So conceptually, the noise, then this natural noise that I have in my synapse is helping me to activate with relatively high probability with low numbers of inputs, okay? This is a very nice idea. So first, dynamical noise amplification. And the second one is very nice, is the topological amplification. And that is mediated with these triangles I was talking about before, okay? So for instance, with this triangle, with this feed-forward triangle here, the activation of this neuron indirectly it is strengthening the activation of this one there, okay? So with these feed-forward loops, I have a higher probability for the system to activate and pass information to other neurons. And these two mechanisms Actually, we explored them in detail, and in these different papers, we demonstrated that these two mechanisms are sufficient for the system to spontaneously activate all the time. Okay, this is the central idea. I don't need anything, and that's why the retina, the spinal cord, and other systems can continuously activate without any kind of effort or dictating neurons to be spatial. Yeah. Okay, this is very nice, and this is an example of how the dynamics looks like. Yeah. While the video plays, something that we realize that is very important is that you have feed-forward loops like this one. You get also feed-backward loops, but in your network, sometimes you need a balance of both of them. The reason is that if you amplify too much the system, it can get saturated. Yeah. If you amplify very little, then the system does nothing. So you need a little bit of balance, okay, for the system to achieve the maximum spontaneous activity level in the network. But depends, of course, of the density of the system, of the wiring of the neurons. But conceptually, my message here is that you can really play a lot with the topology of your system, different architectures, and you can demonstrate that when you have a balanced system, okay, like this one, in which you have different kind of motif, that is the ideal scenario for the activity to initiate and periodically activate again and again your network, as you are seeing here all the time. Okay, these are noise, this is noise, and this noise gets amplified, and finally a bursting event occurs in my network. Yeah. Okay, so this is basically the idea. Something very funny that we observe in these kind of problems is that the activity tends to start in a particular location. Okay, this is something that caught the attention of our team in the sense that if I measure where these activity events are starting in my system, okay, so you have to imagine that I have a noise, the noise gets amplified, something happens, and then activity is triggered in a particular location, and that starts this highly strong propagating wave like these ones, okay? But if you see experimental data and also numerical simulations, you can see, for instance, here with experimental data, that most of the time the activity is starting in this corner over there, okay? And this is because the network that is being formed during development favors the activity that is amplified goes into that basin of attraction, okay? And this is something that we also demonstrated numerically, but the important thing is that if I get this kind of data in which here each dot is the initiation of a particular event, and then the yellow spot marks basically statistically which was the region that initiated the activity more often, okay? And you can see that it changes. When the system is 7 days old, activity is there. When it goes 14 days old, this initiation changes location, and it is because the system is evolving all the time. New connections are formed all the time in my system. And this means that I cannot freeze the network in a particular state. It's impossible to do that so far, okay? And this development, this natural development, is causing this shifting in the initiation, yeah? Another thing that is very beautiful is that if I take a knife and I cut the culture, if I do that, I'm eliminating a lot of these motifs in my system. Many of these triangles are deleted, destroyed, because the lesion in my network. When I do that, what will happen is that the system will continue activating spontaneously, but this initiation area will shift somewhere else, okay? This is very nice. It's extremely hard for me to really stop the spontaneous activity of my system. And this makes neural networks very reliable, okay? Very reliable, and they are able to heal themselves, actually, and cope with damage continuously. Good, okay, so because I like the concept of damage very much, and this is one of the few beautiful things that you can do in vitro without really big revolution in ethical concerns, okay? I will show you an example of how interesting is this. It's not only an interesting, let's say, game for physicists, but actually these kind of experiments provide very nice ideas to understand how a neural network is able to heal itself after physical damage. For instance, during seizures, for instance, or during blood flow disruption in the brain, millions of neurons can die, okay? But the system is able to recover itself and carry out the same functions again, okay? And understanding this problem is very difficult, of course, with humans and with animals, but in vitro provides a very nice playground for testing, okay? So for that, I need a very quick overview of networks, just to show that here I put two typical networks. One is that is highly segregated in the sense that I have very nice communities in my connectivity matrix, and that are represented here in my network map, okay? So the blue is that blue, okay? And you can see that I have very nice communities connected with some connections that are actually putting the different communities together. And it is highly segregated in the sense that I see very nicely the communities, and here is highly integrated in the sense that the communities are very strongly mixed inside the network. They are there, but they're difficult to see, okay? And I put these two scenarios because in network theory, I can compute, of course, the average connectivity of my system. This is low, this is high. I can compute the global efficiency, which captures how easy it is for me to pass information from one node to any other node in my network. So here the global efficiency is low, here is high, okay? But these ideas are intuitive and are very nice and normally help me a lot to characterize the behavior of my neuronal network. Yeah, then I can, of course, quantify how strong is this community segregation. Here is relatively high with this Q value. Here is low, okay? And so on, okay? And also I can characterize hubs. Hubs in the sense that are neurons that mediate the communication with areas in the sense that if these hubs are lost, parts of the network can be totally taken out of my system. So hubs are very important, and they keep the network together, particularly when the modularity is very strong. And with these ideas in mind, what we did in the lab was to first record the spontaneous activity in one of these networks, in this case, the self-organized aggregated network. So you see that activity takes place all over the area, okay? And what we did was to use this data, okay, then compute the effective connectivity using transfer entropy of the communication between the nodes in my system, and then we targeted the node that was more responsible for the flow of information, okay? And this is conceptually is the betweenness centrality. It's a node that supports most of the traffic in my network, and it corresponds to this one over there. And then with a laser, we deleted this node, okay? And then we recorded activity again to see the behavior of my network after the deletion of that node, okay? And then we used network theory to try to understand what happens with my system. So remember, I record activity. I decide who is the node that will be eliminated. With a laser, I eliminate the node, and I record activity again. Then when I do that, after that, what I do that is that I leave this culture back into the incubator for 1 day, okay, to see if it is able to recover or not the previous behavior. And I repeat again and again the process. And this is what I show here. So this is my network map before damaging this node. Okay, this global efficiency is relatively high, 0.42. Then I eliminate this node. It's not there anymore. Okay, you can see it's not there anymore. And then first thing, the global efficiency decreased, okay, by about 10%, something like that. But also local damage that they inflicted here caused long-scale, long-range damage to other effective connections in my network. This means that local damage sometimes can cause effects that are very, very far away and that are very difficult to understand sometimes, okay? So the point is that we have this system, the system got damaged, got affected, it changed the organization of the network, and then I represented this here. So the original network in dark red, then to the damage, the global efficiency goes down, meaning that the system is less communicative, and then I put the culture, this is the networks, then I put the culture back to the incubator, and the system recovers. Overnight, the system is able to recover, so it strengthens the connections that it has, okay, to be able to reach the same activity level as before the original damage. And the reason is that neuronal cultures and many other systems are called homeostatic, in the sense that they try to reach always the same activity level, and they are in the comfort zone, okay? And then what we did was okay, we know that now the system is here, so now we look into the network to the next node that can be targeted and apply damage. And then again, we damage the network again. And then after that, it recovers again, and so on, okay? So we are doing damage, and the system recovers, damage and recovers. And we have to do a substantial amount of deletions, about 8, for the system to really have problems in communicating or showing spontaneous activity, okay? So this is very nice because it's a nice example that shows that neuronal networks, biological ones, are able to recover all the time from damage and try to cope with perturbations. Okay, and then I can compare that with failure. Failure is random damage. Okay, and in this case, you see the system is very, very different, but it also works in the sense that I just pick up one random node in my system, and I deleted that, but the system, again, is also all the time able to cope with this perturbation. Okay? Okay, questions so far? Curiosities? Yes, thank you.
You have on damage step 5, you have an example of the global efficiency rising after deleting a node.
Yes, that's very funny. Yes, we don't know why.
Okay.
We don't know, yeah. Yes, we got this kind of crazy behavior. I mean, for the targeted attack, we got a consistent, actually, trend. But for the failure, we indeed could cause alterations. And the reason for that is that most likely, we damage inhibitory nodes. We damage inhibitory nodes, so the system, indirectly, you make it stronger in communication in the network, okay? And there is a lot, really, at least for me as physicist, this is really a lot of fun to be able to play with these systems and understand what's going on, okay? Good. Any other curiosity? No? Okay. Sorry. If you? The links. So that's a good question, okay. Yeah, it's a good question. The problem is that I don't see the links very well. I mean, I can see the neurons very, very nice, very bright, because when they fire, you see the brightness. It's very easy for me to identify the neurons, but sometimes the connections are so thin that are very difficult to see. And sometimes if I try to delete the connections, sometimes I do a lot of indirect damage to other regions in the network. It's very difficult, because you may think that sometimes you have two nodes, you have one connection crossing the nodes, but sometimes you have a lot of also connections somewhere else that are mixed up together. So it's very difficult for me to see exactly where is taking place. That's why we go into the nodes. A hub, yes. I killed the hub. With some chemical, yes. I can also do that. Yes, I can do that. Oh, sorry. Yes. Thank you. Yes. You can also play. Oh, I didn't try. It's possible to do it. It's possible, but we didn't go into these details of local perturbation. You can apply global perturbation, global, in the sense that you weaken all the connections in your system, or you strengthen them, but to locally block something chemically is difficult, but it's possible, okay? Anyway, thank you, Silvio. I realized that the camera is getting a bit, okay. Okay, so then the last part, the last 5-10 minutes, is that, of course, when I was trying, again, to develop these systems that show rich spontaneous activity, and now, the one I said, spoiler, I don't want to show it, okay? And actually, the idea was to try to make or to emerge this rich dynamic behavior by using, let's say, substrates that are as rich as possible, let's say, in imprinting rich connectivity features in my network. And my students had the idea to use the Sierpinski square, like this, okay, in which we have valleys in neurons. So some neurons will grow in the valleys. So, and these neurons will actually also connect with the top of the profile, but with different connectivity, lower connectivity than with neurons inside each of the valleys. So basically, you are breaking the isotropy of the system by creating regions that have higher connectivity connected with low probability to other regions. Basically, you're enriching as best as you can the blueprint of your system. Okay, and you can see here, for instance, that you have neurons everywhere in the different valleys. And you can see that actually connections are totally crazy. Sometimes they follow the profile. Okay, you have something very, very non-uniform. Okay, and we like that very much. And the idea was to see whether this is enough to make the system very, very rich from a dynamical point of view. And this is what we have. So now I can show the video. So this is the original one, very boring. Okay, and now this is the one with the Sierpinski square, in which you can see that now you have activity in small locations. Sometimes the entire system, again the entire system, and now soon you will see that everything fires except the center, I think. Yeah, now, and then only the center. So now, you see, so you get a very rich repertoire of possible activation states in your network. So conceptually, this is like the mouse thing, the mouse in vivo system that I was showing before, that you have a lot of activations across the network. And we found that very beautiful. And we wanted to explore that in the context of criticality, in which basically you can say that the brain actually is neither in a disordered state, let's say, of random activity, nor in an ordered state in which everything is activating together, but you are more in a critical state in which all possible dynamical scenarios are represented. And to work in this problem, we work with Miguel Ángel Muñoz from Granada, Spain, taking advantage of this recent paper that he published, in which he used the renormalization group approach to model this kind of critical behavior. Actually, to understand this, let me illustrate you that conceptually what we want to achieve is that this kind of behavior is around the critical point in this criticality context. So to understand this renormalization group approach, the idea is that imagine that I have something very simple, like 6 neurons that are here. Okay, so this is my original raster plot. This is my initial group of 6 neurons. Then what I do is that I use correlation analysis to group together those neurons that have similar patterns of activity, okay, like this one. And then I take those neurons with similar patterns of activity, like this one, and then in pairs, I sum up the behavior that they have, okay? So the 2 blues, I sum up the 2 blues, the 2 reds, and the 2 greens, okay? And then I get something like this. And then when I have this data, then I compute the variance of each of these trains, yeah? Then I average, I get a value, and then I repeat again to this next step, okay? Averaging again the next group of neurons, okay? So of course, you have to imagine that you have 600 neurons, so you will go from 600 neurons to 300, those to 150, that to 75, and so on, okay? You're gradually reducing, in groups of 2, your amount of neurons in your system, okay? And the idea is that if my system is in a fully coherent behavior, fully coherent state, like this one, all neurons activate in a highly correlated manner, this variance that I was showing before, okay, it scales with the number of neurons inside this cluster with an exponent equal to 2, okay? This is one of the trivial scenarios, and the other trivial scenario is that when neurons are activating in a completely random manner, there is also a scaling, but the exponent is equal to 1. And the funny thing is that when you reach something in between, okay, you could say that the system is approaching the critical state. Yeah? And the funny thing is, how do you get your neural network to reach something in between here? And of course, once you are there, you want to compare with real in vivo data to see if you're close or not to the behavior observed in the brain. This was basically the idea. So when we put, let's say, the very boring control culture, we got that the system was, of course, with this on-off behavior was in this trivial state with exponent 2, okay, very, very close to it. Then when we go to the fractal, oops. Okay, it decided to die. Okay, sorry. Okay, back to the presentation. Okay, then we are here. This is exactly. Then when we go to the fractal culture, then we are actually already far from this trivial state. Yeah. And finally, when we compare this with in vivo data, we actually get something that is in this regime, which is very close to what we have with our fractal neuronal culture. Okay, so conceptually, we are far from trivial exponents, so we like this idea very much, and this, at the moment, okay, is telling us, or is suggesting us that these fractal cultures are reaching some kind of critical state that is comparable to what we see in vivo, okay? And actually, all these ideas are very nice, and actually we got a lot of inspiration from the work of this work of Mario Capelli and colleagues, okay? And actually these ideas that we are developing here using this renormalization group approach is now creating a very nice collaboration with Silvio and Mauro Capelli that we will discuss that, but we will have exchange students that we will work in the two groups, okay? Silvio and Mauro. And the idea is that we want to keep developing these ideas, and actually they are very, very nice. And to finish this, you may think that, okay, this seems very promising. It seems that with a fractal culture, you can reach a state that looks critical. The problem is that I'm not telling the complete truth unless I put this information here, and is that I can reach that level, close to criticality, only in a small window of time. And it's because the system is developing all the time, yeah? The system keeps developing, keeps making connections, so I cannot freeze the system in this particular critical state, okay? So development will kill the criticality.
And go to the alpha equal to 2 case. In that case, it moves to the alpha equal to 2 case.
Exactly. The system goes into this on-off behavior. And this is the frustrating thing. It's for me very difficult to engineer neuronal networks that have interesting properties, and I keep freezing them in that state, okay? So also, everything depends on the excitation-inhibition balance, okay? If I block inhibition, the system gets totally crazy, okay? And fires too much, and so on, okay? Good. So that is what I wanted to show. Also, for those of you working with criticality and scale and critical exponents, you should know that critical exponents by themselves and power laws are not enough. You need scaling relationships. And with the work of Mauro Copelli, they found that this relationship holds. Okay, so we tried that in our data, and indeed, first, our control scenario is far away from this relationship. It's not possible that it shows critical behavior, sorry. Okay, I bought a new computer, but this computer is not really. Okay, so the important thing is that we really like very much these. Oh, wow. Okay. Sorry. I thought I could trust this new computer. Okay, sorry. It deleted it. Damn it. I have to go back to my presentation. Well, I hope I can finish. Sorry for this. Okay, because I'm almost there. So I wanted just to show that these scaling relationships hold. Okay, very well. Where is the mouse? No. Sorry, thank you.
Be careful with
with these ones, no? Yeah. Yeah, but, okay, thank you. Okay, so just I wanted to tell you that basically the homogeneous culture is very far away from this scaling relationship. This is the experimental data for the fractal case, and these, the black circles, the green squares correspond to the in vivo data, and both experiments in vitro and in vivo, they go correctly into the expected place. Oh, I'm very sorry. I hope I can finish. Where is the mouse? I was very close to finish. Anyway, so it's basically I wanted to say that the scaling relationship is working, and I just wanted to finish the talk because my computer is sabotaging me. Okay, so I wanted just to show you this video to finish my presentation, and this is a video in which people in Cortical Labs Australia was teaching the neuronal culture to play punk. Okay, so basically is the neuronal culture the one that is governing this path? Okay, to play or to try to send the ball back, okay? And the idea is that each time that the neuronal culture was failing with playing with the pad, okay, then stimulation was given to try to rewire the network and try again, and so on. So people was all the time applying different perturbations to try to teach the system to really follow the position of the ball, okay? And another example, let's see if I can show this, is this one in which people is using a robot. This is more sophisticated because here you have to You have a robot, and in the top of the robot, you have a neuronal culture. So the robot is getting, from the sensors, is getting information of the environment. That information goes into the neuronal culture, and the neuronal culture is the one that sends instructions to the wheels to move and try to find the target, which is this object here. Okay? And the idea is that they repeat this again and again, and they apply stimulation to the system. And the idea is to see whether the robot is more and more efficient in finding the target, and you increase the training of the neural culture. Okay? And this is from these people in Japan. Okay? They promise that it works very well. The problem is that all these aspects are very nice, very beautiful, very motivating, but are very, very difficult. It's extremely hard to really train one of these neural cultures to do a particular task and in a reproducible manner. But in any case, for physicists, for electronic engineers, it's a very, very beautiful problem. Okay? And yes, now the computer seems to help me. So now let me just finish with thanking my PhD students that work super hard to make all these experiments, numerical simulations, scaling relationship analysis, and so on. Okay, so Ake and Anna work in this European project. Then Laia, Mireia work in these fractal patterns. Mikkel work a lot in numerical simulations and understanding the communication between the neurons. And Sarah work in the experiments with a laser killing the neurons. And also my set of collaborations that include bioengineering teams in Japan, electronics people in Spain, and also collaborators in physics around the world. And thank you very much for your attention. Thank you. And sorry again for my computer. I apologize. It's his fault. Okay, thank you.
Thank you very much. Time for questions.
I have a question, but I think it's going to be a hard one. What is the role of astrocytes on those cultures? Because I think it can be quite complex.
I didn't mention that. Yeah, you're totally right. They are there. They are present there. Okay, they help the neurons to operate in a healthy way. Yeah, and at least for these experiments in which I'm a physicist, let's say, and I just want to investigate propagation, networks, and so on, I don't really care about the astrocytes. But in collaborations that I have in medicine, okay, there we explore, for instance, cultures in which the astrocytes have a problem, in which they have a genetic disease, for instance. And then we investigate how damage at the glia astrocytes level is affecting the behavior of the neuronal cultures. And we could demonstrate that even if the neurons are completely healthy and they work correctly, because of the astrocytes that are dysfunction, you see a lot of physical damage, actual neurons dying, or making wrong connections because of the astrocyte failure. And actually, we published a paper on that and inviting the medical team to develop therapies to cure those astrocytes in particular. Yeah, but it's an important problem. Yes, I agree. Thanks. I think Dante, no?
Yeah, unless there is a student. I have the following. This is going to be a long conversation, so I'm going to just give you the headlines, and then in private, we may discuss, okay? You mentioned that it's hard to get the system critical, or if you get it in a small window of your parameter or a small window of time and so on. And a few years ago, I mentioned to you a problem that I see in the approach. When you print the interactions, and criticality is a correlation. An Ising model is a lattice, but when you put the right temperature, you get correlation that are the right way, power law, and so on. And it is my impression, and we have modeling published result theory on that when locally you have order, which is your case, even the fractal case, locally is always a lattice with holes, but it's a lattice locally. So neurons have ordered interactions around. Yes. And so in that condition, for K up to some 10 or 20, you have this explosive first order transition and back, first order and back. Okay. But if you manage to put some short circuits, some few shortcuts, a la Watt-Strogast, then the transition is second order. This is in parameter space. You have local order and then the global disorder. So what I would like to discuss with you later is if that could be a hope to get a big, big parameter region in which your circuits became critical a la mouse.
You are right, but it's very difficult. What you are suggesting to create bridges, right? Shortcuts along the long range, let's say shortcuts, it's very hard, at least in culture, because you have to force axons to extend very, very long distances, okay, in paths that are not the same, the culture itself. So you need like a two-layer culture, okay, one with short range or normal range, another one with long range. You're right. We can discuss that. Okay. Thank you. Yes.
Let's go first.
Hi, thank you. When you show the fractals, you have find that the diversity of fractals, like having different
design.
yes, like triangles.
Correct, yes.
The diversity of fractals is
Exactly.
near to the critical state. Have you find a relation?
You are right. They are all very, very close. We tried the triangle, okay, the square, and then the diamond, I think it's called. Okay, we tried different versions, but our experimental approach was not refined enough to see strong differences between the different patterns. They all came. I mean, if I have a culture and I repeat different experiments and I average the results, when I average the results, the standard deviation of my data, okay, then you see that all the different kind of patterns, Sierpinski, diamond, etc., they come inside the same standard deviation, meaning that it's very hard for me to see any strong impact from the detail of the pattern itself. Okay.
Okay, and I have another one.
Yeah.
What is the purpose to train a group of neurons to do an activity? Where, for example, optimize neural networks to deep learning or something like that, or what is the purpose?
You mean to train the network to do a specific task, you mean?
Yes.
Yes, you can do that. You can do that using these electrodes that are around, okay? You can do that. The problem is when you train the system, and then the system, as soon as you stop the training, then the system tries to go back to the origin. And then when you have achieved the training, the system may start activating plastic mechanisms somewhere else to compensate for that training. And this makes things in culture extremely difficult. And I think that the only solution to train a system and keep it in the conditions that you like is that you're all the time feeding the system with inputs, and the system processes something and gives an output, like a real brain. The real problem with cultures is that they are not a brain in the sense that they're not receiving inputs all the time from the environment to take decisions. Yeah? This is the difficult part.
Okay,
yeah.
Thank you.
Thank you.
So what is it about the growth of the mouse brain that stops it from making this fully connected supercritical state? And can you make something similar in these cultures?
Yes, we think that stimulation. If you provide stimulation, pattern stimulation with these electrodes all the time to the system, pretending that it's actual information from the outside, then you can control and keep the connectivity in a state, okay, that is more or less stable. But the problem is that you need to provide stimulation and feedback. So the system has to do something that is meaningful to keep the connectivity in a particular way. But it's extremely hard in vitro. That's why it's a difference with the brain. It's very difficult, yeah. And you wanted to say something else? Oh, no, okay, thank you.
Some more? Yeah.
So thanks for the lecture. My question goes along with the past question. Well, you're cultivating the brain and you do not do any input to the network. So how it grows and why it is growing and why it is activating?
No, but the brain evolves into ways. The first one, genetically, you tell thousands of millions of axons where to go approximately. Okay, so you go in that direction, okay, approximately. And you also move neurons to particular locations. That is the core architecture of the brain. But then as soon as spontaneous activity starts to take place and you are born, then all the information that you get from the outside starts to refine at high speed all the circuits that you made more or less like that, okay? And this is the important thing. In the brain, you get immediately information from the environment to refine the circuits, but in the cultures, that is not happening. That's why we think that you need all the time stimulation or feedback with environment for the culture to keep it doing something specific. These people that used the cultures for the Pong game or to find targets, they claim, I have not seen the data, but they claim that the system is reaching the critical state when they learn properly to play the game, what they claim. And they also claim that you get an architecture of the network that is more or less stable. Yeah. But I cannot promise because I don't have the data. Okay.
Okay. One more. Do you plan to do some experiments with inputs and try to make it learn something?
Yes, we are trying. Actually, we're trying in the sense that we are now buying small robots. So we want to repeat this thing in our lab. Okay, we want to repeat these ideas. That's why I'm now making collaborations with people in electronics, so you can put all the electronics in the robot to, in real time, give information to the neural culture, and that one give information to the wheels or other information of the robot. But we are working on that. Seems fun, but it's difficult actually. You can invest years and get nothing. Yeah, thank you.
I was looking for this, and I found there was a video from a company in Australia called Cortical Labs. And they just claim to have taught a neuronal culture to play Doom, the first-person shooter game.
Yes. Yes. I saw someone send me. Yes, Pablo sent me from Twitter, no X, no? You send me the information. Yes, exactly. I have a collaboration with them actually. We see each other, we discuss ideas, but the work that they do is extremely difficult, in the sense that you need a lot of training. This training is not open thing, okay? It's like a black box. So people is training the cultures, but you still don't know exactly what do you have to do, what particular training protocol you have to follow to exactly achieve a particular action, like a particular game or whatever. It's a bit random sometimes. The feedback is also part of the training protocol. I mean, you have to stimulate, then you get information, then you stimulate somewhere else to try to rewire the network in a different direction, and that is a difficult thing. The entire protocol itself is what people would like to pay millions of dollars to really understand how you should do everything to the system to go in the direction that you wish. It's very hard. Yeah. And that's why things are kept secret sometimes. Yeah.
Okay, so maybe it's time to thank our speaker again.
Thanks a lot. Thank you.